we're here with Dr. Suresh Ramalingam, who's the Deputy Director of Winship Cancer Institute at Emory University School of Medicine, who's going to discuss some of the exciting lung cancer abstracts being presented this year. Thank you so much for dropping by. We really appreciate it.
Thank you for having me, Gina.
So, everybody's talking about FLORA. It's definitely a very exciting trial. It has been from the beginning, and now we've been eagerly awaiting the overall survival data that was presented at this year's meeting. So can you give a little bit of background on FLORA and talk about the OS data that is presented?
Absolutely. So the FLORA study was designed to compare the efficacy of osimertinib to either erlotinib or gefitinib in EGFR mutated patients. We enrolled patients with exon 19 and 21 mutations. These were previously untreated patients, so it's a clean first-line therapy trial, one-to-one randomization, osimertinib or erlotinib or gefitinib. And the primary endpoint was progression-free survival. Noteworthy for the survival analysis is that we allowed crossover. So patients treated with erlotinib or gefitinib at the time of progression, if they had T790, were allowed to cross over and get osimertinib. And we actually had a high degree of crossover in the trial. We've already reported the PFS results in the New England Journal of Medicine. Osimertinib resulted in a substantially improved PFS, 18.9 versus 10.2 months. The drug is approved in many parts of the world, including the US, for frontline therapy. Now we have the mature overall survival data, which is what we presented at this meeting.
Congratulations, it's a very exciting study.
Thank you.
How do these data kind of impact now your sequencing strategies, and what are you doing for patients who progress on osimertinib?
Well, first of all, what we learned at this session is that osimertinib improves overall survival. The median survival with osimertinib was 38.6 months, and in the control group it was 31.8 months. So it's approximately a 7-month improvement in overall survival that was statistically significant, and we can say it's clinically significant as well. We also noted that 47% of the patients in the control group who went on to receive some post-study therapy got osimertinib, which reflects the real-world situation. So clearly, this study met its primary endpoint and secondary endpoint. The question is, some people have said we can give first-line EGFR TKI with erlotinib or gefitinib, and then when they progress, you can sequence them. I think FLORA argues strongly against that. Number one, 30% of the patients in both arms of the study did not see a single post-study treatment after progression. That's because vast majority of them died before they could get another treatment. So for those 30% of the patients, there was only one line of therapy. And that argues for using your best agent first. The second point I would make is not everybody who progresses is going to have the T790M mutation. So if you start off with 100 patients with EGFR mutated patients, and if you all gave them gefitinib, only 1 out of 3 realistically has a chance of getting second-line osimertinib, and the other 2 out of 3 patients are going to miss out. So our argument is that this trial clearly addresses the sequencing issue, and with the sequencing in place, there's a survival benefit for using osimertinib in the frontline setting, and that's why we believe should be the standard of care.
Right. Thank you so much for highlighting all of that. So what's next for osimertinib? Where do you see this agent moving forward in the next couple of years in EGFR disease?
Right. What osimertinib has shown is it's effective, it is safe, and more importantly, it's active against brain metastases. All are 3 important characteristics when you choose first-line therapy. As we look at therapies that the patients will be getting for now years, not months, it is important that it's tolerated well and it doesn't interfere with their quality of life. So osimertinib meets those criteria. Obviously, we're not satisfied with where we are. We need to get better for our patients. We are already working on combinations with osimertinib to improve the outcome, to delay emergence of resistance to osimertinib. We're beginning to understand the mechanisms of resistance to osimertinib so we can develop informed combination therapies. And we're also moving osimertinib to earlier stages of the disease. There's an adjuvant therapy trial in resected lung cancer patients called ADORA that's already completed accrual, and we hope to have the results in the upcoming years. We're doing a study in locally advanced stage 3 lung cancer where patients get chemo-RT and then are randomized to osimertinib or not in the post-chemo-RT setting. So The drug is being tested in earlier stages of the disease. In metastatic disease, our efforts are focused on delaying the emergence of resistance and improving the duration for which patients benefit from targeted therapy.
Wow, those are some great points, and those sound like really interesting studies too.
Yes.
Great, thank you. So moving away from Flora, can you talk a little bit about the BFAST study as well as another trial that's being discussed here?
As we talk about targeted therapies in lung cancer, it's critical that every patient with non-squamous lung cancer get tested for these mutations. Now, we use tissue for testing, but increasingly plasma samples are being studied, and we know that plasma samples can pick up these common mutations. We also know that if you pick up a mutation by plasma sample, you can confidently treat those patients with inhibitors for those specific targets like EGFR, ALK, and so forth. The BFAST study was specifically looking at plasma sampling to see if they can pick up ALK. And what Dr. Gadgil and colleagues reported is a cohort of over 2,000 patients who were screened for ALK in the plasma, and they found about 5.4% prevalence in these patients. And based on that, they gave them alectinib, and the response rate was 87%, which we know alectinib is capable of doing in the frontline setting for ALK patients. So what the BFAST study shows is if you detect ALK by plasma, you can confidently treat those patients with alectinib and hope to see the same kind of benefits you would see if the tissue was positive for ALK. And I think we're going to be increasingly incorporating plasma in our workup because not all patients have sufficient tissue to do the testing. And also, there are certain clinical situations where the tissue is just not available.
Good. Thank you. Now, what about some other lung cancer abstracts that are being presented this year that you think have some practice-changing potential?
Well, immunotherapy is an area where there's a lot of excitement and progress we've made. And I would point out two abstracts that were presented at this meeting. The one in the presidential session yesterday is the Checkmate 227 trial. It also came along with the parallel New England Journal publication. What we learned in this study is the combination of EP and Nivo in lung cancer is associated with improvement in overall survival in frontline therapy. This is the first evidence that EP-Nivo combination improves survival in non-small cell lung cancer. The study is a large trial. Over 1,500 patients were enrolled. We found that when you compare chemo to EP-Nivo in both PD-L1 positive and PD-L1 negative patients, EP-Nivo combination has superior survival. What is even more exciting to me is the duration of response within EP-Nivo stands out when you compare to chemo plus immunotherapy approach. So the ability that this regimen can sustain a response for years is something that I think will help us think through where this fits into the treatment landscape. The tolerability profile was also good, and there were no safety signals with the EP-Nivo regimen. We have to keep in mind that the EP that we use for lung cancer is a lower dose than what's used for melanoma. So with this attenuated schedule, this is a well-tolerated, effective regimen. Now should belong in the treatment landscape in the frontline treatment of non-small cell lung cancer. So that's clearly an exciting advance that we saw. Another study we learned about is the Empower 110 trial, which compared atezolizumab head-to-head with chemotherapy in PD-L1 positive patients. And in that trial, we learned that if you had a patient with very high levels of PD-L1 expression, atezo was associated with a favorable overall survival compared to chemo. The intermediate and low PD-L1 expression patients did not have as significant a benefit. So these results are not very different from what we've seen with Pembro versus chemo, where the highest PD-L1 positive patients had the survival advantage with Pembro monotherapy. So this trial, Empower 110, tells me that atezo can be used in the frontline setting when it receives the approval as monotherapy for those highest PD-L1 positive patients.
Great, thank you. It seems like a little bit more of a trend this year with some chemotherapy-free regimens across different subtypes, right?
Absolutely. And obviously, it'll boil down to what else can we do to increase the proportion of patients that benefit from immunotherapy. What we see is a subset of patients benefit, and they have sustained benefit, and you see the tail of the curve getting better. But we need to see how to improve the proportion of patients who get benefit. So novel combination approaches, looking into biomarkers, are going to help us deliver that next set of progress.
Well, great. Thank you so much, Dr. Ramalingam. This was amazing. Thank you so much for sharing your insight.
Thank you for having me.
Yeah, of course.